Okay, thank you, Peter. All right, so I'm going to be telling you about single molecule electronics, and specifically using single molecule electronics that have been built with proteins. You and I have the opportunity to live through an amazing time when electronics are doing more and more because of scaling. As we scale down traditional electronics, we're able to achieve higher speeds with our computers, lower power per unit of processing. We can cram circuits in with higher and higher density, and even take advantage of effects that really only kick in at the smallest scales, like quantum effects, tunneling, for example. And you and I have seen how this has changed our lives, right? Both with portable electronics and our desktop electronics and all the rest. And a natural progression from this, at least an intellectual progression, is to continue that scaling on down to the molecular scale, to build circuits and transistors and memory bits out of molecules instead of out of silicon and germanium and copper and gold, those sorts of things. And that has led to a field called molecular electronics, which is up there in my title. Molecular electronics is this field of physicists and chemists working in the area trying to answer the questions of, can you build circuits out of single molecules? And if you can, what do they do? Well, it turns out that during the time of this development of molecular electronics, you and I have been able to buy handheld terabyte drives and gigabit networking and small flash drives that are based on flash memory. So in fact, we don't need to work at the molecular scale in order to achieve high speed, low power, high density, and quantum devices. And for that reason, the field of molecular electronics today is relatively bankrupt. That particular field, trying to use molecules as transistors, or trying to shrink electronics to the point of single molecules, is at a relative dead end. And that's because molecules do not conduct electricity. Molecules, for the most part, are not transistors. They are not memory bits. Now, molecules do store memory. Molecules store tons of information in really interesting and fabulous ways, but not in the ways that an electrical engineer would think of as being a memory. Now, my group works in a corner of molecular electronics. Our goal is to produce circuits out of molecules in order to make circuits that are chemically functional, to take advantage of some of these more complex behaviors, what real-life molecules really do in nature, and try to merge that with solid-state electronics in order to do something completely new. Not just a bit, not just a transistor turning on and off, but to truly merge the function of chemistry and biology, chemical biology, with processing power, traditional memory storage, and worktop computing that you and I have all come to know and love. All right, now this is not a field per se. There aren't very many people trying to achieve this sort of thing. And the last time I was here speaking to the faculty a number of years ago, it was still just a premise. It was not demonstrated anywhere in the scientific literature. What I'm going to tell you about over the course of the next hour or so is the progress that we've made along these lines, actually trying to achieve these goals. And the first step is to use circuits that are small enough that you can connect up into these molecules. So if I draw it, in principle, it's relatively simple. I want to be able to measure voltages and currents, and I just want to measure these electrical signals at this single molecule scale. And that's going to require building pretty small circuits. The purpose, just to state it again slightly differently, is to watch molecules in action. To watch molecules attach and bind and recognize each other and dock to each other and pass information and energy back and forth between them. All the complex things that happen in biochemistry, to watch those processes happen molecule by molecule, atom by atom, in real time, using the power of something like a nanocircuit. Now, Peter introduced me by saying I've been working with carbon nanotubes for a while now, and you can imagine carbon nanotubes as being one platform for doing this. And it's not necessarily the only platform. but it's an example of how you might merge a traditional solid-state electronic platform with these sorts of gooey, very non-solid-state systems in order to do real-time electronic recording of the processes of life, of dynamic and complex chemical events, and to do that with single molecule precision. All right, so in my group, we actually do start with carbon nanotubes. Our particular carbon nanotubes are called single-walled carbon nanotubes. So I've got the acronym there, and we grow them ourselves in our lab. We synthesize a little iron, that's supposed to be iron moly, Keggin nanocluster that acts like a seed that sprouts the nanotube, and we do it on silicon wafers, and we do all the traditional patterning, and I'm not going to really tell you about any of that, even though it eats up an enormous amount of our time and graduate student effort in order to make this all work correctly. In the end, we produce circuits that look something like this, where we can run current through a carbon nanotube. And there's an SEM picture of what one of these circuits really looks like. The source and the drain are off in the corners of the image. The nanotube is the kind of diagonal white line that goes wisping through the image. And what we do with most of our circuits is we cover all of the circuit in order to protect it from the environment, either using, well, here I've drawn it using a polymer called PMMA. Also, we'll use an oxide, like aluminum oxide, in order to protect the device from the environment so that it's not interacting with the environment. And then we'll open up a small window and expose just a section of this. So you can think of this as a transistor that's about 1 nanometer wide and a few hundred nanometers long, exposed to the environment, the air or water or whatever liquid you might dunk it into. And the advantage of the PMMA or the other passivation is that the rest of the circuit doesn't short out when I go and I put this into a drop of blood, say, or a drop of some other electrolyte. All right, now, one of the main focuses of our group has been to then take this platform and figure out ways of attaching single molecules to it. And we, over the years, have come up with 2 different schemes. One is a covalent scheme, where you're making a covalent bond, and the other is a non-covalent scheme. And the covalent technique takes advantage of a defect on the nanotube. We start with pristine nanotubes. We introduce a defect electrochemically, and once there's a defect there, depicted here as a carboxylate, then we can do chemistry that only attacks that defect, essentially attaching things to that scaffold without changing the rest of the nanotube. And so here are some images from a few years back showing molecules that were attached using this defect scheme. In this case, the nanotubes, again, are the white wisps running horizontally through the images, and the bright dots are single molecules of streptavidin that have been chemically attached to the side of the nanotube. And you see we got pretty good at getting just one of these attached to each of our transistor circuits. This we've studied for a number of years, and a number of the students in my group, Steve Hunt and Tatjana Sheps, have studied what happens when you introduce these defect sites into carbon nanotubes. It's particularly interesting from a physics point of view because you're introducing a scattering site into what would otherwise be a one-dimensional channel. But we don't use this technique very much anymore. The pictures are very pretty, but they're hard to make. It's not really scalable. So we could make one of these per day, but you couldn't imagine making thousands of them at a time. It's not really practical if you were interested in perhaps commercializing this. And so a few years back, we switched over to a non-covalent technique, which has actually been really nice for us to work with. It's much more general, and it's something that we can reproduce much more readily. And so that's the technique that I'm going to tell you about mostly today. Here, we're not putting a defect in the carbon nanotube. We're not damaging the carbon nanotube. We're just sticking molecules down to the surface of the nanotube. We stick it down using this molecule that I've labeled pyrene maleimide. The pyrene maleimide molecule has 2 parts. One looks like a group of 4 benzene rings. And those 4 benzene rings act like Scotch tape on the carbon nanotube. They have the exact same atomic registry. They form a nice van der Waals bond to the carbon nanotube, which is also made of these carbon hexagons. And so the pyrene end of this molecule sticks things down to the tube. And the maleimide group Well, malamides are a good anchor group for attaching to sulfurs, thiols in chemistry, and specifically cysteines. So a cysteine is an amino acid, it's one of the building blocks of proteins. And so I can get a protein to attach to my pyrene malamide anchor as long as the protein has a cysteine in it. So here now is a much higher resolution image of one of these sorts of devices. Now you see we're resolving the nanotube. This is an atomic force microscope image, so I'm zoomed much closer in. And with this kind of image, we can measure the topography very carefully. And so I can tell you that the carbon nanotube there is exactly 1.8 nanometers in diameter. And the protein, the little blob that's attached, is 5.2 nanometers in diameter. And that corresponds to the size of the particular lysozyme protein that we attached here in this case.
How do you get one defect on a nanotube?
Well, in this case, there's no defects. But how do we get one molecule? I'm going to tell you that. So first off, we use these pyrene anchors using a combination of concentration and pH and then rinsing. We can get a very dilute coverage on our transistor. Remember, we only have a few hundred square nanometers of surface area that we're working with at the very start. And then with the dilute coverage, we might only have a dozen or so of these anchor molecules. And then the attachment of a cysteine to the anchor molecule, that's also a low yield reaction. And so if you have a few dozen of the anchors, you might only get a couple of the molecules attached. And so there's a statistics to it. We tend to work in the limit where there's either 0 or there's 1. So that it's very clear when you get one of these proteins attached, like in the image again, that's another one, so the sizes are a little bit different. Rather than, of course, you could crank up the concentration and these molecules would be all up and down the transistor, but then you'd be back in ensemble physics where you'd have many of them interacting and behaving independently. We tend to dilute the pyrenes, we dilute the protein until we have just one of these attached to our chip. And then what we found is that we can dump this into liquid, into salt buffers, we can dry it off, we can image, we can measure it some more. And these attachments are very stable over days and even weeks. So the same device, the same molecule, can be measured day after day after day until ultimately we get tired of it and throw it in the trash and go and do it again with another one. In this case, the drawing is of a particular protein. It's T4 lysozyme. And I note the mutation. We've actually introduced a cysteine where we want it on the protein, so that every time we attach a lysozyme, it attaches in the same orientation with the same, at the same binding site, because there's just that one cysteine that will react with our linker molecule. And then once we've created a circuit like this, we might measure it in different solutions. This is the type of signal that I'm going to be talking to you about today. This is a plot of current on the y-axis versus time, and it's just 1 second of data, just a snapshot of the sort of signal that's generated in the nanotube as lysozyme works away on its partner molecule, here labeled peptidoglycan. So as lysozyme goes and captures peptidoglycan and releases peptidoglycan, each time that happens, we get one of these pulses above the baseline current. And of course, we do all of the necessary controls. We change the concentration of peptidoglycan, we remove the lysozyme, we've used mutants of lysozyme that don't have this activity to prove to ourselves that these signals really are the chemical processing. And I'll talk more about that in a moment. All right, but here's the outline of my talk. I've kind of been going through the motivation of this work and then the fabrication, how it is that we make these circuits. I told you just real briefly about the two schemes, but we've come upon a chemistry that's relatively flexible and allows us to apply this technique to a wide variety of different things. We're limited now only by manpower rather than the technology of putting these together. And what I'd like to do next is tell you about work that we've done on 3 different enzymes. Lysozyme, protein kinase A, and DNA polymerase are 3 very, very different proteins that we've attached to these carbon nanotube circuits and then studied in order to understand a little bit better. And I'm going to start with the lysozyme one. All right, so lysozyme is the soldier of your immune system. You have it in your tears, in your saliva. It's the way you and I fight off bacterial infections. Lysozyme is like a Pac-Man. It opens and closes, and that's why I had Pac-Man on my opening slide. It opens and closes to break apart peptidoglycan. And peptidoglycan is a polymer that forms the cell wall of certain kinds of bacteria. So a cell is a bacterium is enclosed within a cell wall. That cell wall is a polymer of peptidoglycan. And you and I have evolved this special protein, lysozyme, that's very selective. It goes and attacks the peptidoglycan. It rips the peptidoglycan apart, ultimately splicing open that bacteria and killing it. One of the nice things about lysozyme is that it has been studied for over 100 years. And it's been studied by single molecule techniques. So science, the scientific community, has a pretty good idea of exactly what lysozyme does, what sorts of mutations have different effects, and what its rates are. So we took lysozyme as a good starting system since there was all that existing data. Now here are 3 examples of the kinds of signals that we see when we're monitoring lysozyme with an electronic circuit. Sometimes, and I'm sorry, I've changed my accent. So here now I'm plotting changes in conductance rather than current, but it's the conductance of the transistor. It's still the current that's flowing through the transistor from source to drain. So sometimes a lysozyme just sits there and does nothing. It doesn't generate any signal at all. Other times, the lysozyme opens and closes so quickly that on a 1-second time scale like I've got here, you can't even resolve the individual events. But if you zoom in, we can very, very clearly see that this signal is still made up of high events and low events as the molecule, as the lysozyme closes down upon the substrate and opens back up. We're collecting these signals at about 100 kilohertz rates. And today we're collecting them closer to megahertz rates. And so we're generating many hundreds of thousands of data points with approaching microsecond resolution of these molecular events. Now I said there is an inactive type of lysozyme. There's this blue type of lysozyme where it opens and closes very quickly. There's also an activity where the lysozyme is opening and closing much more slowly, only about 10 or 20 times per second. And because people have studied this with single molecule optics, of course they've observed the same sort of thing. Some molecules do nothing. Other molecules are opening and closing super fast, and it turns out that that means that there's no chemistry going on. And other molecules, the effective molecules, the ones that are actually doing the catalytic processing, those are the ones that are moving slowly, only about 10 times opening and closing per second. Now, because of the high quality electronic signal, we can go in, we can measure the amount of time that it spends closed, we can measure the time it spends open, we can generate probability distributions of both those times. You see that the distributions here are nice and linear on a semi-log plot. It's just a very simple Poisson process with a single time constant. And from these distributions, we can pull out time constants. So for example, the lysozyme spends about a quarter millisecond in its closed conformation, and it spends about 3 milliseconds open before chomping down on the next bridge in the peptidoglycan. And that's what gives us a rate of about 330 hertz, at least here for the blue data, for that ineffective and fast binding. The green data has slightly different timings, of course. It spends a little bit longer closed, about 3 or 4 times as long. It spends a much, much longer time open, 64 milliseconds, and that leads to a much lower, slower terminal rate of only about 15 hertz. All right, so far, everything that I've told you was known about lysozyme. It was known that different molecules have these different behaviors, and people had watched it using light-based techniques. Nobody had ever watched a molecule before using an electronic technique. So in itself, this is interesting that we're measuring current on our y-axis instead of measuring photons on our y-axis or force on our y-axis, the way others do in the single molecule science community. But what's more, our devices are extremely stable. We can watch a molecule. We can watch a molecule all day if we like. Usually we don't because the hard drives fill up and then the computer crashes. Our problem is always the storage capacity. Here's a color-coded plot of 3 minutes worth of data now. The molecule is attached to a transistor. We're measuring the transistor current. We can do that as long as you're patient enough to wait. And here we're seeing something that was completely unknown before about lysozyme. A single molecule changing gears. A single molecule. going back and forth between this fast blue behavior of being ineffective and this much slower catalytically active behavior of breaking bonds. And even stopping for a moment to rest in its inactive conformation. Over the course of minutes, you can build up excellent statistics on the mean time that it spends in its active behavior, or the mean time that it spends in this behavior that's too fast. And in fact, the mean time, you can read it right off the plot. It's on the order of tens of seconds, or 10 seconds. There are times here when the molecule spends 30 seconds in one particular behavior, a behavior that's governed by time constants on the millisecond. And then after half a minute of doing that, it suddenly changes gears and starts doing something completely different for the next 10 or 15 or 20 seconds. So here we've combined something that's really unique. Really good time resolution down on the microsecond scale with really good stability on the minute and even the hour time scale. So that we can watch not just the individual chemical events, but the buildup of a whole ensemble of chemical events as a molecule goes through the whole process of its lifetime, all the different variable things that it can do.
Well, Aaron, how can you be sure that it's the molecule that's changing and not the nanotube on the longer time scale?
There is there are changes in the nanotube. There's backgrounds there, and we measure that, and that background has been subtracted in this case. The nanotube does change with characteristic timescales, and we're aware of that, and we have to correct for that. That's a good point. I should point out all these graphs that are plotted around 0. That's 0 nanosiemens away from some baseline, and the baseline has a wavering behavior to it.
What's that other color?
The other color. So let me see if I can explain this. It turns out that lysozyme can be totally quiet in 2 different ways. It can be stuck open, and it can also be stuck closed. Since we have when we have a well-defined 0 level, we can distinguish between those 2. And so here I've colored in black places where the molecule is stuck open, and that only happens in these green periods. And I've also separately colored places where the molecule, for whatever reason, got stuck in its closed conformation. Now, normally it would only spend a fraction of a millisecond in its closed conformation. Here, every once in a while, we see that it's stuck in its closed conformation for multiple seconds in a row, and that only happens when it's in this ineffective binding gear. So it's kind of got a first gear and a second gear, and sometimes it gets stuck. Okay? And I've drawn you a little pie chart there to break down the amount of time that just one molecule spends in these two different gears, or maybe stopped at the stop sign.
What gets it unstuck?
We have no idea. How it gets out of it, and what kicks it from one gear to the next, that's still well, we have guesses, but it's somewhat unknown. There was another question?
Yeah, what is the time scale that other measurements are stable at? You mentioned that your method is much more stable than other methods. How long can you measure it?
So the standard in the field is a fluorescence-based measurement, where you're looking at the blinking of a fluorophore. You shine lasers on it, and then the light that comes out indicates whether the molecule is closed or open. And the problem there is that under laser illumination, those fluorophores bleach, and they bleach in just a couple of seconds. So you can get pretty good resolution of molecular motion at millisecond scales, but then after a couple of seconds it goes dark, and you have to go finding another one. And that's what precludes being able to watch a molecule for 10 or 20 or 30 seconds and see these different transitions. So in most single molecule techniques, you're only able to watch a molecule for a few seconds at a time, and then you have to move on to another. Now we've tried to vary the environment of these. Since the circuit is stable, you can imagine changing the salt concentration. Here I'm showing you a table of changing to 3 different pHs, and we've also done some work changing temperature, so you can ramp the temperature up and down. It turns out that some of those measurements are extremely difficult, and frankly they're better suited to working in a beaker, because I can measure the overall turnover rate of lysozyme versus temperature just on a hot plate if I want to, and I just turn it up and turn it down. But our hope is that we start to see more information at the single molecule scale by playing with these different variables. And so, for example, look at what we saw by varying pH. At pH 7, where lysozyme is in U and I at pH 7, lysozyme spends only about half of its time actually doing processing. So you could think of its efficiency as being about 50%. And as you go below 7, either down to 5 or up above 7 to 11, you see the efficiency drops off pretty radically, down to 10% or so. The pie charts provide a nice graphic representation of some of the things that are going on there, as this very, very fast, ineffective opening and closing grows at expense to the catalytic processing. But look also at what happens to the inactive time. The inactive time on average only lasts for about a second or two at pH 7, but it skyrockets up to 10 or 20 seconds when I go off pH. So the presence of those extra protons or protonated groups causes lysozyme to get stuck for much, much longer times and spend 20 seconds at a time just doing nothing, being completely ineffective. All right, I'd like to move away from lysozyme now to a second molecule that we've looked at. I'm going to tell you a little bit about a molecule called pKa, or camp-dependent protein kinase. pKa is the traffic cop of all of your cellular signaling. It's the protein that turns on other proteins and turns off other proteins. So pKa is really critical for starting and stopping other processes when your cell needs to, either everything from something really simple, such as I've received a photon, I've recognized light, to it's time to go. This cell needs to blow up and kill itself. So pKa does this. It does its regulation in a slightly more complicated way than the lysozyme data I was just showing you, because pKa binds two partners. It binds two molecules. It binds a molecule of ATP, and then it binds a substrate molecule, a protein, and in our measurements, the protein that we've been using is peptide. And then mechanically it brings them together and transfers one of the phosphates from ATP over to its substrate, to the protein that's its target. And then it opens up and it releases those two in order to start the process over again. So just like lysozyme, it's catalytic. It's not being consumed in the reaction, but it's changing one protein from some native chemistry to having an extra phosphate group. And then that extra phosphate is what activates that protein or deactivates that protein, depending on the particular signaling pathway that we're talking about. So pKa is really critical. There's been almost no study of it at the single molecule scale. So unlike lysozyme, there's a lot that's unknown about this molecule, and every measurement that we do uncovers new information. Again, we've taken the native protein and we've introduced a particular mutation. The C here stands for the cysteine. We're still using the same scheme of putting in a cysteine so that we can attach it down to our carbon nanotube. Here's what one actually looks like. So there's a carbon nanotube, WISP, again going through one of the AFM images, the cartoon of what that might look like, and then here's now what the data looks like. It's a little bit more complicated. It's not just two levels. It's not just open, close, because now in this case we have two binding partners. And let me show you that in a little bit more detail. Here's current versus time for about half of a second for the carbon nanotube with the protein attached, sitting in a salt buffer. Now this particular protein, pKa, needs to bind to a magnesium ion. So the salt that we're using here is a magnesium chloride. And here now this is an absolute current scale. So you can see these are all the fluctuations in the system, including the underlying fluctuations of the nanotube, and the electronics, and the fact that the lights are on, and the doors closing, and the protein itself. The protein's random motions are all incorporated in the width of that histogram there. Now if I add a little bit of ATP to the solution, now my histogram of the current breaks out into two clearly defined states. I still have the same baseline, but now I have current drops on the order of 5 to 10 nanoamps, which indicate the binding and the unbinding of that one partner molecule, ATP. Now here we're not doing any chemistry yet. pKa doesn't change ATP, it's just binding to it and then releasing it again when it's not in the presence of other proteins. And so we're going between two known conformations, and again, if I zoom in on any one of these individual events, you can get some idea of the quality of the data there. Here, you're actually seeing, let's see here, the bandwidth is probably only about 40 kilohertz or so, right, Patrick? So you're actually seeing a roll-off here, which is not the protein motion, but just bandwidth limit. Now that's with the ATP. If I bring in both a combination of ATP and that target protein, the substrate protein, then I get not just the intermediate conformation, but also this closed conformation. And I've zoomed in on one of them down here in the bottom right-hand side in order to give you some idea of the classic catalytic cycle. We start with the open protein here in blue. We bind one partner or the other. We don't have any control over that. And then as we bind the second partner, that initiates the closing of the protein and the bringing together of those two molecules. So we go from the open state to an intermediate state, we close up, and then it pops open with the successful release of now the phosphorylated target protein, the phosphorylated
substrate.
substrate.
The open close valve?
Where does the
valve, which you said closed and open, so there must be a valve or some connection. Where do I see it on the picture?
Yeah, well, so there's certainly a hinge. The molecule has a hinge right about here, and it's the top half and the bottom half that are coming together. The control mechanism, the valve that you're asking about, well, it's driven by the energetics. So the binding of a partner here causes one conformation to be lower in energy, and then the protein starts to move towards that conformation. And the combination of both binding events causes this conformation to be the lowest energy, and that's what then brings the thing together. Of course, all driven by room temperature thermodynamics. All this has to work inside you or me, and so it's not connected up to a battery or anything like that. Okay, so there's a single typical cycle, and I heard another question.
Yeah, I assume that when you introduce this mutation within the cysteine, that does not affect the function.
Yeah, that's a good point. Anytime you mess with a protein, you have to go back and double check that you haven't changed its native behavior. Surely proteins were never meant to be attached to nanotubes, and we can't control for that. But as far as we can tell, the average activity that we see is the same activity as you would have for the native protein in solution. Or if there is a slight change, we measure that. Our collaborators, Greg Weiss and his group in chemistry, are really good at that, and we work very closely together on this. But that's a good point. When we attach, we have to worry about how much of the behavior we're perturbing. All right, so let me show you something interesting that we did with pKa. I already showed you this typical single cycle of capture and closing and then opening back up. But every once in a while, we see cycles that are much more complicated, where we get that capture event, the enzyme closes because it drops down to its low current value. And then as it starts to open back up, it snaps shut again. And it opens and it snaps shut. And it snaps shut multiple times before finally being able to open all the way back up. Now the biochemists have studied this using techniques like NMR, and they tell us that if the protein cannot open until the substrate has been successfully phosphorylated. And so the fact that it's repeatedly trying means that it's repeatedly bringing these two molecules together and trying to get that phosphate to transfer over. Until the phosphate transfers over, it's unable to open its mouth again and take another bite, just like you and I when we're eating peanut butter. Okay? So this thing is chomping away on these two things, trying to get them to stick just right, and has to open up a little to allow them to move around, to sample a slightly different conformation before bringing them together again and seeing if it can't initiate that reaction. Now, this is a this is a case where we can watch a molecule for long, long periods. We've watched hundreds of thousands of events from a single molecule. And therefore, you can build up a really nice plot of the probability of even these rare events happening. The simple cycle, just the classic one, that's what we see 77% of the time. 12%, 5%, we see it taking 2 closures or 3 closures in order to successfully transfer that phosphate group. But every once in a while, you get one of these really rare events where perhaps it takes 12 or 13 or even 14 closures before that transfer finally goes over. And you see that there's 2 different slopes here, or at least there's a nice clean slope here in the bottom half of the plot. So that suggests a constant probability. It's just trying over and over and over, and knows it'll eventually get there. And then the curve, this plot curves up, suggesting that there's something really highly probable about making this successful on the very first try. Now let me try and put that into the context of what we know about the ensemble rate. Here I've plotted frequency, so the number of events per second, and the dashed line is just what's known from a beaker measurement, where an ensemble of these molecules are transferring phosphates with a whole ensemble of target molecules. And if we look at our data second by second, that's what I'm plotting here at 10-second averages, we see that a single molecule might run at the ensemble rate for 10, 20, 30 seconds, and then suddenly change gears up to a completely different rate, 4 or 5 times faster than the ensemble rate. So in the first half minute, the molecule seems to be doing exactly what you'd expect from the ensemble measurement. But in the second half of the minute, it now seems like it's mutated. It's done something very unusual. It's going much faster than it should be. If we zoom in on the actual activity during these 2 sections, well, it turns out that when it's going slow, it's actually being very inefficient. These slow periods are when the molecule has to close multiple times in order to perform one of these transfers. And over here, the ensemble rate goes up to 80 or 90 hertz when it's being very, very efficient and transferring it every single time on the first try. And the abrupt change from one type to the other, well, it resembles lysozyme to some extent, where lysozyme had these 2 different gears of operating. Here we're watching pKa operate inefficiently for tens of seconds, and then very, very efficient for tens of seconds, and back and forth from minute to minute. And that's a thermal fluctuation of some slight conformational change of the protein as it samples all the different possible conformations that are still active. All right, we've also worked with DNA polymerase, and polymerase is a critical enzyme for building DNA. It's the way that you and I reproduce our DNA when we make new cells. And now I'm thrown off because so that clock is not real time. What time is it, Peter? Is it 4:00? It's 4:15. Oh, okay. I'm pretty good. So let me. So I do have just a couple minutes. Let me just give you a couple of highlights of DNA. DNA is interesting, of course, because you can't open the paper these days without hearing something about DNA diagnostics and the coming era of DNA in healthcare and sequencing DNA and all the rest.
Neanderthal DNA.
Neanderthal DNA, right? And yeah. There's so much public interest in DNA as well. Well, so polymerase is the molecule that helps to build DNA. And specifically, it's the molecule that turns single-stranded DNA into a duplex. So if you have a duplex, first the duplex is separated into 2 single strands, and then polymerase comes and binds to one of those strands and builds it out by finding the right complementary nucleotide and forming the different base pairs. So again, I'm going to show you plots of current versus time. Here I chose a time axis of 10 seconds, just randomly. We can measure polymerase attached to one of these circuits with nucleotides, without nucleotides, with single strands of DNA, with double strands of DNA, and all the different combinations. And the important point here is that these circuits have some random noise to them. In this particular case, the random noise is plus or minus 4 nanoamps. But when we're actually doing the process of building a strand, so in this case, in the middle plot, it's poly DC, so it's C, 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 in a solution that contains the complementary G nucleotide. So C's do not bind to A's. The C's don't do much of anything when they're just in buffer. But a strand of C's in the presence of the complementary nucleotide generates all this extra electrical noise, all these extra electrical switches. And it turns out, again, if you zoom in on these, you can resolve them really nicely. And what we're seeing now is the catalytic cycle. As polymerase works its way down the strand, reads the strand, says, aha, I've got a C. Looks through the solution for the right binding partner, aha, there's a G. Brings it in, and then ratchets its way along to the next base in the strand. Let me describe an experiment for you. Imagine that we take our device and we provide it with just a very, very low concentration of these strands. So low, I think here we're working at nanomolar, that one of these strands only lands on the device once every 5 seconds or so. You would expect the current to be very, very quiet for many seconds at a time until that binding event happens. And then polymerase goes to work. Step, 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 step along the strand until it comes to the end. And that's exactly the sort of thing that we've seen. By working at very low concentrations, we're able to see these long quiet periods followed by the capture of a strand, multiple of these events, and then the release of that strand until it waits for the next one to come along. It's diffusion limited. We're just working in the limitation where diffusion only brings us a molecule once every 10 seconds or so. We can count up how many of these excursions. In this case, if you see up at the top, I'm using a 42 C bases. And in fact, I measured 42 of these excursions. And here's a sense of the histogram that we built by looking at these clusters of excursions and counting them up. Excuse me. So sometimes we see that the polymerase goes for 20 or 30 bases and then falls off. But many times we get exactly 42 of these excursions, which means that we go all the way down the length of the single strand before kicking off at the very end. All right. In summary, this has really been fabulously fun. We're learning a lot about these different enzymes. It's at the interface between physics and biology and also solid-state electronics. And in each of these different publications, we're learning new things about the protein function that were never known before, even on some of these proteins that have really been studied for a very long time. And with that, I want to say something in just the last few minutes about the device physics, a little bit more about exactly how these transistors are actually working. Yeah, question?
I was just wondering, in our cells, how fast polymerase moves? How many bases can it do?
About 15 to 20 bases per second. Okay. And we're seeing exactly that. 15 to 20, it incorporates 15 to 20 bases per second. Yeah, there was another question.
So the example you showed, you had the C and you had the G coming in. And can you differentiate between different nucleotides?
Not yet.
Okay.
Yeah, so we have tables. So we've done all the 4 different bases and all the 4 combinations and the 16 different mixtures, and we've looked at the timing of them, and we can't read out CG, CG, A, T, T, T. Not yet. But when we can, then some of these guys will be able to graduate.
So you got a random solution of strands of C's, and a lot of the G stuff around. So basically, whenever the polymerase wants to stick a G on, there's one available.
Yes, that's right.
Lots of G's.
And the concentrations that we're using are the same kinds of concentrations that would be in your cell. So your cell produces G's and also C's and A's and T's. So at a certain concentration to allow polymerase to do that job.
So your thing has to sit around and wait for the C's, but the G's are almost instantaneous because there's so many?
Yes. In this experiment, we use an excess, so we use like a biological concentration of the G's. So there's plenty of G's. The diffusion time for those is a nanosecond.
Okay.
And then we used a tiny, tiny concentration of the C's so that we could resolve these events and separate.
So you get these separate clusters, but the clusters are at the rate that the molecule wants to work.
Yes, that's correct. Once one of these has been captured, then the other binding partner, there's plenty of that around, and so we can just run it at full speed. That's right. Okay, let me tell you a little bit about what we've done in terms of understanding these devices and where the signals come from, because this is really critical to reproducing it and then being able to generalize it to whichever protein that we're interested in. All these molecules are being measured in a salt solution. And salt solutions, electrolytes, have a screening length. And so it was very natural to ask, how does the electrostatic screening in a salt solution affect the signal that we're measuring? Here are some experimental data from a lysozyme device where I plotted on the top some different salt concentrations from 10 millimolar out to almost 1,000 millimolar, and the corresponding Debye length for electrostatic screening of each of those different concentrations. Now, we found that the strength of our signal falls off perfectly according to a standard screening kind of Debye-Huckel model. And there's only one free parameter in this. So it tells us that, sure enough, Debye-Huckel screening is critical here. And we solve for the parameter, and the parameter is 1.0 nanometers. Now that parameter tells you how far from the nanotube is the charge that's being screened. And we had thought that the charge we would be screening would be 4 or 5 nanometers, because that's how far away the binding site is. The pocket where peptidoglycan binds, up here in the middle of the protein, is 5 nanometers from the nanotube. The whole protein is 7 nanometers tall. And yet Debye-Huckel tells us that the thing that's actually being screened is only 1 nanometer from the nanotube. So we zoomed in on this region that's within 1 nanometer of the nanotube. It turns out that all these proteins are known with atomic accuracy because they've been crystallized and studied by X-ray crystallography. So we know the exact position of every atom when the protein is closed, and we know the exact position of every atom when the protein is open. And we know which of those atoms have net charges on them. Well, it turns out that down here at the base of lysozyme, where we're attaching to the nanotube, there are only 2 positive charges. Everything else is net neutral, at least on the outer surface of this protein. There's only 2 positive charges that have any significant movement. Here I've drawn in 2 different grays, I've overlaid the X-ray crystal structures for the protein open and the protein closed. And so you can directly visualize the motion of charges, and there's 2 of them. So we said, oh gosh, you know, maybe this signal is coming from just the motion of these 2 protons being waggled around. It's called an allosteric motion when one part of a protein moves in response to a binding event somewhere else in a protein. So lysozyme has this allosteric motion. The amount of motion is known, and gosh, even a good Physics 7 student can calculate the change in electric field on the nanotube if I have 2 protons that move by 2 certain distances. Of course, it's a vector problem, and so it takes them all weekend rather than just a couple of minutes. But it's a relatively simple physics problem. And so we dreamed up an experiment where we asked, what if we mutated the protein? What if we knocked out this positive charge and made it neutral? Could we change our signal strength? Now notice, where we propose to do this knockout is far from where the peptidoglycan binds. It's nanometers away from where the peptidoglycan is being broken catalytically. And so it's reasonable to assume that we're not going to change the chemistry of lysozyme by changing something down here. But of course, we do have to actually go and test that. Well, it sounds like such a good experiment that we went wild and we turned it into a 9-month research project. This required an enormous amount of work by some of the people in my research group, in combination with Greg Weiss's group again. We synthesized 7 different variants. We took the native one, which had these positive 2 charges. We knocked them both out to make the tail negative. We inserted negative charges to make both charges, we made both charges negative, and then we did all the other combinations as well. One positive, the other one negative, etc. In order to build proteins that were positive, negative, and neutral. Then we attached each of these to nanotube transistors. We measured it, we threw it in the trash, we grabbed another one, we measured it again, so we measured multiple devices with multiple different variants, and then put together an entire table of the results that we saw. And here I'm just going to summarize that table for you. The bars on these represents the strength of the signal. And so with our variant that has the 2 positive charges, we get a nice strong positive signal. The variant that has the negative charges, we get a almost equally strong negative signal. The variants that have 1 charge, either one way or the other, are almost the same and half as big. And the variant that has no charge has almost no signal. It's symmetric if I subtract away a certain amount of signal from everything uniformly. And we understand what that means. It tells us that the whole protein is made of charges, right? The whole protein has all of its movement. And that whole protein gives us a certain amount of signal. It's our baseline signal there, which here on this plot I've labeled as 34 millivolts worth of signal. And then in addition to that baseline from the whole protein, there's extra signal coming from these charges that are so close. And those close charges are the only ones that we're mutating. And as we mutate them, we can drastically increase the signal, either positive or negative. And what that shows us is that this is sensitive to as few as 1 charged amino acid. We can design a protein with just 1 charged amino acid, or you could go overboard and put in 2 charged amino acids and produce these nice rich signals that I've been showing you throughout this whole talk. So now we have a system. We have the cysteine attachment. We know how to generate signals. We can design these charges, these mutations, far away from the catalytically active site so that we're not interfering with the native activity of the protein. And we can design in the size of signal that we need for a particular background in order to generalize this to any other kind of molecule. With that,
you're also saying that you have to have a charge that's close to get good signal to noise.
Yes.
So there's some, it's nice that the ones that you happen to have a charge that's really close.
Well, we get to choose, so let's see. So you have the protein structure, and hopefully you know where the chemistry is happening, and we get to choose where we're going to attach that protein to our nanotube, because we get to choose where we're going to put in that cysteine, and we can choose whether we want the neighboring amino acids to be charged. And so that gives us a lot of flexibility for how we want the protein to attach to the circuit, and then which part of the protein movement is going to transduce an electric signal. And we're testing that now. We've taken the same protein with 2 or 3 different kinds of attachment sites in order to measure, well, how does this part of the protein give a signal compared to this part of the protein, and do they have different kinetics or do they match? All right, so that's the summary of my talk, a little bit at the beginning about why we want to do this work and how we're actually achieving it. I've told you a little bit about 3 different kinds of enzymes, all very interesting proteins, all with completely different functions. And then I cut short here a little bit, but a little bit about how these transistors are working to give you a sense that we really can design around them, and therefore we can scale it out and basically attack just about any biochemical problem that we're interested in. And with that, I'd like to thank you all for your attention, and also, of course, those who have been contributing to this work, both in my group and Professor Weiss's group. It's been a 5 or 6 year effort, and so lots of people have contributed, and they all deserve thanks for this work. All right, thank you.